Hi, my name is Natalie Lobby and I work with Dr. Leah Michelle in her research lab. I will be presenting on my summer research on gamma D crystalline protein interactions. I'm going to start out with some background information about my research. So what are crystalline proteins? Crystalline proteins are proteins in your eye lens that maintain lens transparency, and there are three types. The three types are alpha, beta, and gamma crystallins. Alpha crystallins work as chaperone proteins to maintain the interactions of your beta and gamma crystallins and keep them functioning properly. Beta and gamma crystalline proteins maintain the transparency of your eye lens by maintaining the structure. We study gamma B crystalline, which is the bovine homologue to human gamma D crystalline. And to the left, you can see an image of what gamma D crystalline looks like. So why do we study crystalline proteins? When crystalline proteins stop interacting correctly, it results in aggregation or phase separation, and these things can lead to cataract formation. So as you can see in this diagram to the left, when your eye lens stays transparent, when your proteins are not clumping together or binding to one another, but when they start forming aggregates, it can lead to cataracts. So what are cataracts? Cataracts are a medical condition in which your eye lens becomes cloudy, causing light to be scattered or blocked as it enters your eye, which can impair your vision and cause problems. Why are cataracts a problem? There's no actual cure. The only option if you get cataracts is to have a surgery in which your eye lens is removed and an artificial eye lens is put in place. But the problem with this is that cataracts can reform in your artificial lens and it is inaccessible in some parts of the world. Cataracts are also a problem because they affect a huge part of the population and they will affect most people at some point in their lives. Congenital cataracts are also a leading cause of treatable childhood blindness, which is another reason why cataracts are a problem. So what causes cataract formation? When crystalline proteins stop interacting correctly and their interactions are increased. And this can be caused by UV damage, old age, mutations, or smoking. So our research focuses on studying gamma B crystalline protein interactions via NMR. When we do NMR experiments, we do T1 and T2 experiments. These experiments provide us with a rotational correlation time, also known as a tau C value. A bigger tau C value means that the protein is rotating slower. So when we obtain our tau C value, we know that if the proteins interact with each other more, bigger molecules will exist in our samples, which will cause the molecules to rotate slower, giving us a bigger tau C value. So our plan for the summer was to purify wild-type gamma B crystalline for NMR experiments, purify the D107A crystalline mutant, choose another mutant to study, and optimize our methods along the way. The mutants that we study are point mutations. This means that between 1 and 3 nucleotide bases are substituted for different ones. This results in a different amino acid in the protein's primary structure, which can change how the protein interacts and other aspects of the protein. The first mutation we are studying is the D107A mutation. This is a homologous mutation to the E107A mutation in the gamma D human crystalline protein. This mutation is similar to the wild type in structure, stability, and solubility. It's unstable when mixed with alpha crystallins, and it is associated with congenital cataracts. To the right, you can see the glutamate, or the E, that will be substituted for an alanine, or A, at position 107 in human gamma D crystalline. This mutation is also unique because it causes the isoelectric point, or PI, to be raised by approximately 1 whole pH unit. The P23T mutant is the other mutation that we have decided to study this summer. This mutation also has a similar structure to the wild-type protein. It is also linked to multiple forms of congenital cataracts, And there is no clear mechanism for the significant decrease in solubility that we observe. In the image to the right, you can see where proline P is mutated to threonine T at position 23. And in this diagram, the mutation has already occurred, and this is a threonine. So our methods and results. Our general methods to prepare for NMR experiments are to obtain our gamma B crystalline protein through recombinant protein expression in E. coli, to purify our protein using size exclusion and then ion exchange column chromatography. And then we normally have to repeat steps 1 and 2 several times because we need such high concentrations of protein in order to perform our NMR experiments. So once we have enough protein in order to do our NMR experiments, we perform T1 and T2 NMR experiments to probe changes in the protein-protein interactions. Once we've done all the NMR experiments we want to do on our wild-type protein, we'll create our desired mutants using a quick change kit and site-directed mutagenesis. Then we will go through steps 1 and 2 again, obtaining it through recombinant protein expression, purifying with size exclusion and ion exchange column chromatography. And then once that's all done, we can perform NMR experiments on our gamma B mutants and then compare that to our wild type NMR data. So as I mentioned, one of our goals for the summer was to optimize our methods along the way. And one of the ways we optimized our methods was by changing our gels that we run for SDS Page gel electrophoresis from 10% bisacrylamide gels to 12% bisacrylamide gels. And our overall goal was to better separate the bands of smaller proteins because crystalline protein is a smaller protein. So as you can see from these pictures, in the 10% gel on the left, it's hard to distinguish what is protein and what is the sample buffer that's run through the gel. But to the right, you can see a clear band where our crystalline protein is, and you can easily see the size of it because the protein ladder has run all the way down. Another way we optimized our methods was by introducing a low pH precipitation, which allowed us to increase our protein yield by almost 4 times. In this method that we introduced, we add HCl to our lysed cells, which precipitates out proteins that are unstable at low pH. Our gamma B crystalline is stable at low pH, so it is not precipitated out. Once we have our precipitates in solution, we centrifuge it down and we are able to pour off the liquid that is left, which will contain our protein, and we're able to discard those proteins that precipitated out. So our current work. To the left is an image of an initial HSQC NMR experiment that we performed last week with our wild-type crystalline protein. We have wild-type protein that is folded and ready for further NMR experiments that we will be performing in the upcoming weeks and months. We also are planning on performing light scattering experiments to be done on the same exact samples we are doing NMR experiments on, and those should be done in the coming week. Our D107A mutant is in the process of being purified, and our P23T mutant is in the process of being prepared. So what's the broad impact of our research? Overall, we want to gain a better understanding of the mechanism behind cataract formation. And with this better understanding, it could one day lead to the development of a prevention method for cataracts or the creation of a cure for cataracts. So what are we going to keep doing in the future? We want to perform more NMR experiments. We want to study more gamma B mutants linked to congenital cataracts, and we want to study the interactions between gamma crystallines and alpha and beta crystallines instead of just the gamma-gamma crystalline interactions. I'd like to acknowledge the RIT Honors Program for providing me this funding for the summer and allowing me to do all this research. I'd like to acknowledge Dr. Michelle and her lab group, Dr. Thurston and his lab group, and the DRIG funding source that Dr. Thurston has that allows us to do this research. Thank you for watching.